Okay, our topic for this video is to look at tools that we use for identification. And specifically, although an expert may need to use identification tools on occasion, we're really concerned with tools for the person who is not the expert, who needs some guidance in how to work their way through all of the characteristics of organisms in order to figure out who they are. So I guess the first question would be, well, maybe it's obvious. Why do we need to identify stuff? Well, we need to, this may seem, as I said, obvious to you, kind of straightforward, that we need to identify things in order to understand the characteristics of the organisms that we have. And so this comes into play in, agricultural pest management, in quarantine, being able to identify whether an insect that's found in a shipment of grain coming from, I don't know, Brazil, is a potentially dangerous pest or not. Comes into play in questions of conservation. How do we know which species are rare and which ones we need to take measures in order to conserve, if that's what we decide to do? Identification is key to these and many, many more things. And many people, I think, believe that taxonomists exist for the purpose of identification. Now, don't get me wrong, there are some positions, professional positions of identifiers. And to fill those positions, typically the people that are hired are taxonomists, because a taxonomist has built up some expertise in a group, or at least expertise with the techniques of identification and the tools that are used, the tools that are available. But these jobs are few and far between. There's a systematic entomology laboratory in Washington, part of the USDA, APHIS, whose job is identification. So quarantine officers will send their questions to them. California has a similar sort of system. And Florida has a similar sort of system. So areas where we're getting a lot of importation and quarantine is an issue that you have people whose jobs are to identify insects. But the vast majority of taxonomists are doing other things. They're working as conservationists. They're working as university professors. They're doing many other things. And so while you may think that because someone has expertise in a group, that they are willing and able to provide identifications. That's not necessarily straightforward. It may be true on certain occasions, and of course it also depends on the particular question you're asking, whether or not it's an easy question to ask, because sometimes, even for the specialist, identification is a very time-consuming and difficult process. So a lot of the chores or task of identification falls to the person who needs the identification. And in this sense, the research that a taxonomist does, usually one component, you go in and you make hypotheses about what the species are and how they are separated, and you provide a way for others to use that information that you have discovered, to use that information in order to make your own identifications. Not necessarily because I don't have the time or the inclination to do an identification, but everybody is a finite individual, and you want to pass that information forward to the next generation. So developing tools for identification are very important, and knowing how to use those tools and what went into constructing them, and how to carefully use them and effectively use them is a real skill set that I think is being lost these days. So you've talked about some of this in the lab this week, but let me go over it again for you. The standard tool to use for identification is something called a dichotomous key. the idea of the key, meaning that's the key to unlock the knowledge, in this case to arrive at an accurate identification. And the term dichotomous simply means to cut in half or to cut in two. So a dichotomous key works very similarly to, perhaps you remember the game called 20 Questions. There used to be a TV show called What's My Line, which was kind of along the same the same idea, where on What's My Line, the mystery guest would come in. Sometimes it was a recognizable person. They'd have the panel with blindfolds on so they couldn't see them. And the panel would ask questions of this mystery guest in order to arrive at an identification. I think 20 questions is probably a little bit more apropos because in 20 questions you have a limited number of questions to go from a universe of possibilities down to a specific identification of the object in question. And so if you do it well, at every step of the way, you're going to go from a universe to half a universe to half of that to half of that, and thereby home in on the identification. That's basically the way a dichotomous key works. Excuse me. In structure, the typical structures you're going to come across in the English language literature, or at least native English language literature, not stuff that's been translated, as we'll see in a second. The key is divided into a series of numbered couplets. And by couplet, I simply mean that there's two halves at each of these numbered steps. The halves of a couplet are sometimes called leads. Although you don't really hear a lot of people using that term. They'll talk about the first half of the couplet or the second half of the couplet. And within each half, or within each lead, you are presented with a series of characteristics. And basically it's a question. Does it have this, that, and any other thing? If so, do this. And if not, then go to the second half of the couplet, check and see if it fits there, and if so, do that. So as an example here, I've got a very simple-minded example here. You come into the beginning of a key, and number one means it's the first couplet in the key. Sometimes you'll see 1' for the second half of the couplet. Sometimes people will just put a 1M dash or something like that. In this case, I only have a single character. Wings present. If you look at the specimen that you have in hand, and by golly, there are wings on it, then you go to couplet number 2 and continue the process. If, however, wings present is not true, and in this case I've worded it since there's only one other logical possibility. Well, there is another one. Hang on, I'll get to that. Wings absent. If the wings are absent, then go to couplet 45 and continue the process. The other logical possibility that I just thought of on the fly is, I can't tell if it's got any wings or not. What am I supposed to do then? Well, in the case of a dichotomous key, if you can't tell if it's got any wings or not, and wings is the only character here, you are basically up the proverbial creek without a paddle. There are two ways to deal with that. One is to close the book and walk away and say this specimen is unidentifiable. The other is to take your specimen and continue through both halves of the couplet. So maybe it had wings. Let's go to couplet 2 and try to keep identifying it from there. And if you're lucky, as you continue down what is the wrong path, you'll come up with a series of characters where it just can't be the specimen that you have. And so you can say, oh, well, it must be that there were wings on that specimen, or there weren't wings originally on that specimen. Anyway, that's the usual structure that you'll see. Couplets numbered. Sometimes when you get into the body of the key, it's very helpful, and this is something we tried to do in your textbook, is to include in parentheses after the number the couplet from which you came in order to get to this couplet. So let's say instead of 1 here, it's 41. And here at the 1, it says 41, open parenthesis, 25, close parenthesis. That means that we arrived at this couplet by coming through couplet 25. This can be helpful when you're identifying things, and either you can't get through the couplet, the key going straight forward from 1 to the highest numbers, ending up with identification, but you have an idea what it is, and you just go to the identification and try working your way backwards to see the characteristics and make sure that the specimen you have that you think is this taxon has those characters. A little tickle in my throat. The same logical structure, but a different physical layout, is used in publications that come out of, primarily Russia or the Soviet Union, if you're looking at old stuff, or in some publications coming out of Eastern Europe that were under Soviet influence back in the day. Maybe you can't read Russian, and I could have sworn that I closed that parenthesis there. Maybe you can't read Russian, but of course there's lots of English language translations of Russian literature, so that it may be relevant to you after all. And the structure you'll see that I'm presenting here is exactly the same logical question. Are the wings present? Do this. If the wings are absent, do that. But it's just laid out a little bit differently. You'll come into the key and it'll say 1, and then there's this 45 here. Let's skip over that for a second, and then we see the characters that you're supposed to compare to your specimen. If your specimen has wings, then good. You continue on to number 2. If your specimen doesn't have wings, then that's where the 45 comes in. You go to 45 and down there to, I don't know if you wouldn't really call it a couplet, to lead 45. You see, here's 45. It came from number 1. And here's what the character is, wings absent. All right, I don't have wings. Go to 46 and continue on. Same basic idea. It was really intuitive when I first ran across them. I didn't recognize it at first, but it became very quickly very obvious what you were supposed to do in order to run through them. So it's just the standard dichotomous key. So there's some best practices that a writer of a dichotomous key should do in order to facilitate identification. I guess here I want to stress that in writing a key, the goal is to help the user get to a correct identification. I've seen some systematists claim that they want their keys to reflect the phylogenetic relationships amongst the organisms. And I suppose that could be, you can set yourself any goal you want, but that's definitely has to be a very secondary goal. And it's problematic, in my experience, in almost all cases, because there are always exceptions, or the characteristics that show you the phylogenetic relationships are rather arcane or hard to see or hard to interpret. We want to make the identification key usable and accurate. So, some best practices. If you can, when you're writing a key, the ideal is to have each taxon come out in only one place in the key. This can be more difficult than you might think. If each taxon comes out in only one place in the key, that means that you've defined how many couplets there are going to be in the key. So for n taxa, there are going to be n-1 couplets. If you have a key that has 10 couplets and every taxon comes out once, that means you know that there were 11 taxa there. Conversely, you can have these huge keys. The key to Coleoptera is going to be huge because there's so many different families and there's so much diversity within some of those families that there's simply no good set of characters that will be true of all the specimens you might run across. Why is that? Well, you may have differences between the sexes, that one sex may have wings and the other sex does not. So obviously, if you need to use characteristics of wing venation, you're going to have males and females coming out in two different parts of the key. That's going to inflate the number of steps in the key. And you get to this, you want this balance between characters that are obvious and easily interpretable versus inflating the key to unimaginable sizes. You know, if you're doing a key to the families of beetles, you don't want to individually key out every species of beetle. You may end up being able to know that what you have in front of you is a chrysomelid, but you really didn't need to go through all the pain and agony of distinguishing your chrysomelid from the other 100,000 species there might be. Something else that I strive to do is to facilitate, as far as I can, the user to get to the answer they need efficiently, as quickly as possible. This again is one of those balancing schemes. Logically, you would say that the most efficient way to construct a key is that, say you have n taxa in your that you're trying to key out, and that the first couplet will, the first half of the couplet will lead you to n over 2 of those taxa, and the second half of the couplet will lead you to the other n over 2, so that each couplet divides the number of possibilities in half, homing in on the correct answer. That would be logically the most efficient way if you're pulling out a species at random that needs to be identified to get to an answer by going through the least number of steps. That, however, can be a trade-off. There can be a trade-off because obviously some species are much more common than other species. You know, through very rare things. So you might want to construct your key such that the common things, the things that the user is most likely to have in their hand, come out very quickly in the key, come out very early in the key. And these rare things, okay, maybe you got to go through a few more steps to get there. There's a trade-off there. And often you are also constrained simply by the characters that are available to you. That in order to have easily recognizable, interpretable, visible characteristics, you have to stray from what might be a theoretical ideal structure. Again, emphasizing again that the overriding goal, the reason that we're writing these keys is to make it easy for you to correctly identify the specimens that you have in hand. Other best practices, the leads are symmetrical. By that I mean that if I suppose I should also add into that my example on the previous slide, wings present versus absent. If the character is not visible or is lost, or who knows, for whatever reason you can't determine which half of the couplet your specimen should go through. If there's only one character, then you're stuck. So it's good to have multiple characters to provide supporting evidence, and to have the most definitive ones show up early in the couplet, and the more, well, you know, we might say this is always true, this is true 85% of the time, this is true 50% of the time. So you'll often see characters modified with words such as usually or mostly or commonly or something like that. So in each lead, it's good to have multiple characters to provide corroborating evidence that that's the choice you want to make in the couplet. That raises a problem that I always check for when I review taxonomic papers that have identification keys in them, and that is each lead in the couplet symmetrical? Does each half of the couplet talk about all of the same characters. If I say wings present, tarsi 5 segmented, and cerci elongate, go to couplet 2 versus wings absent, again, you're back in the same point. If you don't have wings, you're stuck because you don't know what the number of tarsal segments or what the shape of the cerci are in the second half of the couplet. So always, a good key will always be symmetrical, be balanced, talk about all of the characters in both halves of the couplet. Another thing to avoid, not always possible, is what I call A, not A couplets. Sometimes you'll come to a point where of all the species left that you think that the user might have, that there's one that's just so weird in so many different ways that the key writer will basically describe that taxon with all its characters: wings present, tarsi 5 segmented, mandibles black, crown of thorns on its head, who knows, all these other characters. And if you go to that, that's a Dibibliomoctus flavipes. But the second half of the couplet will say, not as above. Not an ideal sort of characteristic, but something I know that you will see in keys sometimes, simply because you want to get rid of that very distinctive thing and get down to the core, difficult to identify things. And then the final best practice that I've got here on my list of things is that the characters are definitive, that they are the two halves that are presented in the two leads of the couplet. The differences in the characters are clear, obvious, easily recognizable, difficult to be mistaken for. Again, all ideals that we strive for and reach our goal imperfectly. But that's the kind of thing that makes for a good key, a high quality key, versus something of lesser quality. Okay, so that's dichotomous keys. You'll be using lots of dichotomous keys. You become very familiar with them. Some years ago, we had a visitor from, where is he from at the time? I believe this was David Yates, who is now at the Australian National Collection for CSIRO, the Commonwealth Scientific and Industrial Research Organization in Australia. I think David was a postdoc at the time. Oh, I remember. He was a postdoc at the American Museum in New York. And he had been working on a project with his Aussie mates and came and was showing us this. And basically, they were developing the software to construct what are called multi-entry keys. And I'll always remember that one of the things that David pointed out as seemingly a truism is that keys, dichotomous keys of the day, keys are written by people who don't need them for those who can't use them. That is, keys are written by these experts who know the group inside out. So they really don't need the key to identify the species themselves. But they provide it to you, the user, who have no familiarity with the group, no particular experience with the range of variation to be found, and the terminology is almost invariably arcane and hard to understand. So couldn't there be a better way to do this? And one of the issues here is that the specimens that you have may not, for who knows, a million different reasons, may not be ideal for the way that the key is written. For, perhaps the first character talks about the number of tarsal segments on the legs. And if your specimen has had all the legs broken off, or the legs are folded under the body and the tarsi aren't visible, or the tarsi are embedded in glue and you can't count the number of tarsal segments, then you've got a problem. So they devised a way, and there are lots of other parallel efforts to do this, but this particular project is still ongoing and reasonably popular, I think, around the world. To get around this problem, they found a way to get around this problem. This is called multi-entry keys, and the idea is that in a dichotomous key, the only entry point is couplet 1. And if you're stymied by couplet 1, then the rest of the key is of absolutely no use to you. In a multi-entry key, you can get into the process of identification in many different ways. The product that David Yates was promoting, and I mean, he was just the salesman at hand, if you will, at the time. There's a lot of people involved in this. And it was headquartered at the University of Queensland. There's a product called Lucid Keys, and there's a website called Lucid Central, and the URL there, which I encourage you to try out. There's an online version of the Lucid Key. Not all Lucid Keys are online, but of course they all involve software, and you'll see why in just a second. I encourage you to try it out. I'm going to walk through it. I have a series of screenshots rather than actually doing it live, because I don't know if it would actually work in recording a PowerPoint presentation. I'll have to try that after I get done here to just see if it could actually work. But let me walk you through this key. This is a key to the orders of insects, a Lucid key that you can run online. So if you go to that website, here's what you're first presented with. Basically, it's laid out in 4 boxes here, and of course you've got scroll bars at the sides here, so you can tell that there's stuff down below. At the box on the left-hand side here, features available. So these are characteristics of insects. And the box on the right-hand side is called entities remaining. Since we've just begun our use of this key, all entities are remaining, so they're all up here, all in their classification. They have 31 orders of insects / hexapods. So we can, let's say we have a specimen in our hand, we can identify this specimen starting anywhere we want. That's the essence of what a multi-entry key is. So let's pick, I think I started out with, what was it, 4 legs. Right, so you click on 4 legs, and that option opens up to show you 3 character states. And you need to read these, look at your specimen, and see which of these options your specimen matches. Are the 4 legs armed with stout spines and modified for seizing prey? Or are the 4 legs not modified for seizing prey? Or are the 4 tarsi with the basal segment swollen, containing silk glands? As a critic, I might say that I would prefer that this description of the 4 legs have mutually exclusive options. Obviously, theoretically, you could have the 4 legs armed with stout spines and modified for seizing prey, and 4 tarsi with basal segment swollen. The lucid structure, though, actually would allow you to go both ways. That you'll get to the same answer no matter what you choose, if it's true for the specimen, because all of these taxa over here have been scored for all of these characters. And basically, you're asking the first of your 20 questions here, and it's going to divide these guys out into smaller groups. So let's say it's armed with stout spines and modified for seizing prey. Select that and see what happens. Oh, no, we're not going to do that quite yet, because let's say I don't understand what that character means. Over here, there's a little icon that you see that if you click on it, allows the designer to add images to that character state so that the user can see that and get some guidance as to what the words mean. Of course, this is not a feature that's limited to multi-entry keys. You could do that also with dichotomous keys. In order to have it pop up, of course, your dichotomous key is going to have to be in an electronic format, but you could do that same thing. So this is not unique to Lucid or multi-entry keys, but it's very, very helpful. And so here we see that modified, armed with stout spines and modified for seizing prey. Of course, they're looking at a praying mantis, but it gives you an image of what they're talking about here with the foreleg both labeled and highlighted in red color. Now let's select that option. I've clicked on that button, and now you see that these two boxes have shifted. That now 25 of the orders of insects have no species that have forelegs modified for grasping prey, but that there are several, 6 orders up here that they do have forelegs so modified. And then the next step, this is showing you who they are. The next step would be, okay, let's pick another character. Wing texture. Forewings leathery, hindwings transparent, both pairs of wings membranous of similar texture covered in microscopic hairs. Forewings hard, opaque, forming close-fitting covers over the abdomen. Select one of those and see if your number of choices diminishes, because obviously you want to get to the point where there's only one entity remaining, and that's your identification. I don't recall which one I picked. Let's see. I picked forewings leathery, hindwings transparent. So now I'm down to just praying mantids and true bugs. And you just continue this process, going through it until you hopefully get to the point where you can have only a single entity remaining and you have your identification. And so wing number, I think wing number is not going to help us there. We would need something else. Oh, I picked absent because this is a contrived example. I know there's some praying mantids that are wingless and have the legs and such, and so this will get you there. I think there are also some true bugs that are wingless but have the forelegs modified for catching prey. I will have to tell the developers about that. But anyway, I've come to my answer. And just as I had icons, buttons over here to bring up pictures to illustrate the character that I'm talking about, here for the taxa, you see there's a similar icon. So if you press that, we actually go to an information page about the order what are they calling it? Mantodia. It tells you something, it gives you some description, tells you about their life cycle and importance, and gives you pictures. The Lucid software allows you to make these things extremely elaborate and extremely information rich as you go through them, combining the identification functions of that multi-entry key with basically a little blurb, much as your textbook does. Except it's not something that well, I suppose you could go through the alphabet here and look at each order there. It's not as easily as just sitting down and reading the book. One other thing I wanted to point out to you about Lucid keys. We go back to the beginning. Up at the top here, there's a whole series of icons, and if you're live, you hover over these and they'll give you a little hint of what they are. But you see this one here that kind of looks like a magic wand. What that does, interestingly enough, is that if you press that, the software will consider where you are in the identification process right now, and will suggest to you what the best character would be to look at next. And so if we, at the very beginning of the key, I was surprised at this, if you click on that, it suggests that the tarsimeer number is the best character to get you to your identification most quickly. Would not have guessed that. I would have thought it'd be something, maybe on mouth parts or something along there. But it's suggesting the number of tarsal segments for you to pick to get to the next available answer. So I kind of like lucid keys for lots of reasons. I think you can even construct these on the fly. That was one of the dreams I had at one point, because we score characters for we do our taxonomic work in a database application, the description part. So we have a taxon by character matrix, and we're saying what each taxon has in terms of the characters. That's the basic input that goes into making a lucid key. And so I always thought that it would be a straightforward manner to take that same matrix and output a key automatically along the way. We haven't done that. There's been some issues along the way, but the logic, I think, is straightforward. So which is better, a dichotomous key or a multi-entry key? Well, the lucid folks would argue that a multi-entry key is better because aside from simply by its very nature being a database application, and therefore you can build logic into it. You can have the program do things like say, look at this character next, because it's going to get you to your answer the fastest way. But they would also argue that the ability to first go to something that's very obvious to you. Maybe there's a color character there, and you've got a black specimen, and these things come in all different colors, but not all of them are black. And so if black is obvious, black is definitive, you click black, and already you've shortened the list that you have to get to. So it allows you to use the characters that you understand, that you can interpret, that you can confidently score. Whereas in a dichotomous key, you need to learn what the characters are and what the differences are between them sometimes. And that can sometimes be difficult, especially when we get into very minute or very arcane characters that people don't come into the process particularly understanding. And that's one of the processes of learning to use the keys, is to learn what the characters are and how the person who wrote the key expresses themselves here. And that's all good. And that's all good. I have to tell you that I think there's still a place for the dichotomous key, and that is primarily because I believe dichotomous keys are very valuable. Not only, maybe they're not as efficient as multi-entry keys in getting you to the right answer. Maybe I'm stepping on my own ultimatum back there at the beginning, that the purpose of the key is to get to that accurate identification. But I think dichotomous keys also serve a pedagogical value. It helps you to learn the characteristics and how organisms are subdivided. You sit down with a key, for example, you sit down with a key to Diptera and you start keying out flies for any length of time, and pretty soon you've got the first few couplets of that key memorized. You already know that I need to look at this character first, then this character, and then this character. And by doing that, I've subdivided the world of flies into just this little chunk, and now I can concentrate on that little chunk. And so you learn that. I don't see that being as easy to do in the context of a multi-entry key, where the first character you choose, that black color that I used as an example, is totally trivial. It'll get you to an answer, but it doesn't lead you to any deeper understanding of how the characters are distributed amongst this taxon as a whole. So I think keys are very valuable as pedagogical structures. And I should probably add that to my goal of keys as identification tools. Now I can already hear it. You have a question. Why don't I just use Bug Guide? Why don't I just go to the Bug Guide website there at bugguide.net, got a specimen in hand, and I look for it. I find a picture of it. Or if I've got some it's not a container ship sitting in the port waiting for an identification. I've got some time to get the identification done. I just take a picture, post it on Bug Guide, and say, okay, world, okay, community, tell me what this thing is. Well, obviously, you can do that. It's my background that I prefer to understand why I got to the answer I did, what the characters are that tell me that my identification is correct, rather than, well, if I come into as a totally naive user, I don't know what the important characters are. So perhaps I'm looking at a picture and I'm focused on totally trivial characters. For example, whoops, there we go. These are pictures from Bug Guide. These are actually 3 different species of the common name is bluets. So this is the genus Enolagma in the family Synagrionidae. 3 different species, all males. And I've got, let's say I've got one of these damselflies in hand, and I say, okay, which species is that? How do I know what to look at? What are the critical characters here? I'm frankly hard pressed to identify any characters by which these 3 things differ. I'm told that the amount of black on the abdomen is helpful. So these guys seem to have very thin black, whereas this kind of goes commonly, uniformly around the end. These apical segments, this is, oh, I can't, let's go from the beginning. 1, 2, 3, 4, 5, the 6th abdominal segment. The dorsum of it is completely black, completely black. Ooh, there's a little blue stripe at the beginning there. Is that an important character, or is that a character that varies widely within a species among individuals? I don't know. I'm relying upon these photographs, these identified photographs, to tell me. And of course, if these photographs are identified, that means somebody with the knowledge of the characters has done that identification. So we still needed that taxonomist along the way. An example I'm more familiar with, since I'm not an expert on at the species level for these damselflies, is one of these little wasps. Now you can see at the breadcrumbs up here that this thing has been identified as arthropod, hexapod, insect, ants, bees, wasps, Hymenoptera. Parasitic Hymenoptera, the parasitoid Apocrita. We'll talk about that business when we get to talking about Hymenoptera. The superfamily Platygastroidea, the family Platygastridae, the subfamily Telenomini. All right so far, but that's wrong. That is not Trissolcus. I know that because I've been looking at these guys for more than 40 years. So I can tell at a glance that this is not a Trissolcus. There are some that I can't tell. This is a Telenomus. And I could tell because of the shape of the body, the relative width here, the shininess of the face here, and things like that. But you have to consider the sources here. And actually, if you look at the history of identification of this picture, you see that at one time it was placed in Telenomus, then somebody who thought they knew better, thought they knew right, changed the identification to call this a Trissolcus. So Bug Guide has errors, of course. And while any identification, there's a certain error rate. And ideally, you would either have somebody with the chops that knows the group inside and out, years of experience, decades of experience to identify this thing. You may have to pay for that expertise. Do the identification. You'll have the next best thing is probably a recently published, lavishly illustrated identification key with supporting information for characters that don't show up, in your 20 questions sort of format to do that. And then as you go back in time, the latest knowledge is not incorporated. It was difficult to publish images, even in the 1940s and 50s, they had relatively few images in publications. They happened to be line drawings. It wouldn't be nice photographs, et cetera, et cetera. So the quality diminishes. My criticism of Bug Guide from the point of view of a professional entomologist is that I think it would be preferable for you are going to be professional entomologists, knock on wood, if you don't know the characters and how to identify them by sight, at least you know how to get to that point, how to discover what the characteristics are and how to interpret those characteristics. And that really is the whole point of your textbook, to provide you with a resource to get to the level of family in terms of identifying the insects of the U.S. and Canada, basically America north of Mexico. So that's my criticism of Bug Guide. So let's change tack a little bit and get into some of those papers that you read for this video, talking about DNA barcoding and the pluses and minuses of DNA barcoding. First of all, though, why does the issue of DNA barcoding even come up? And this is a topic that is, well, it's enshrined in the, the Convention on Biodiversity, I guess it is, the Rio Treaty, that talked about the taxonomic impediment. As I began this whole video, I talked about the importance of identification is. And how we get to that identification is we need the expertise of taxonomists, maybe not to identify for us, but to be out there discovering and conveying to us how to identify things. The taxonomic impediment itself is represented by the knowledge gaps that we have. There are relatively few taxonomic impediments in this sense in things like birds or mammals. All right, there are a few birds and few mammals every decade or so that are described as new. But in comparison with arthropods and insects, where there's just huge gaps, where the taxa have not been studied for decades, perhaps ever, or for which there is no trained taxonomist anywhere in the world. These are significant gaps, and in the spirit of you can't conserve it if you can't conserve something if you don't know it's there in the first place. The existence of this taxonomic impediment makes it difficult for us to appreciate, makes it impossible for us to appreciate what is out there, to figure out what's more valuable and less valuable. I personally think that everything has value, but to prioritize our values and then prioritize our efforts to conserve that biodiversity. If we don't know it's there, it'll never be preserved, except by total accident, right? Why is there a taxonomic impediment? Well, one thing is that the number of taxonomists with expertise is decreasing in the world. Like it or not, in some ways, I feel that the field that, let's put it this way, the practitioners of taxonomy and systematics that were around when I began my graduate school career are as rare as hen's teeth now. The science has shifted to now we're getting more phylogenetic analysis, use of molecular data for phylogenetic analysis, things like this. The part of the core, the discovery and description of basic biodiversity remains, but now there's other tasks going on there, and people prioritize their efforts in different ways. So the amount of expertise is decreasing. And concomitantly, if there's not as many people doing this sort of job, that means that not as many jobs available for the next generation to move into after they complete their degrees. So not only are the number of qualified individuals decreasing, the number of jobs are decreasing. Departments are focusing their attention in other ways. In the early 60s, which is before my time, I think at Ohio State there were, let me count them, in the Department of Entomology, in terms of arthropod systematics, there was Dwight DeLong, Don Borer, Ralph Davidson had some expertise there. I'm forgetting somebody also with that expertise. And then there was Barry Valentine in the Department of Zoology. So there were like at least 3, 4, maybe even 5 insect taxonomists in those 2 departments on this campus. And now there is one. I should not. I forgot to count the acarologists too. So not 4 or 5. There were 6 or 7 arthropod systematists, and now there's 2 at Ohio State. So we have contracted that number. As the job description of a systematist, taxonomist has changed through time, as I described, phylogenetics, molecular data and stuff, the amount of funding that is available to do the alpha taxonomy, the basic biodiversity discovery and description, discovering new species, describing them, naming them, the amount of money available is decreasing. And of course, it used to be possible in the old days that you didn't even need to get grants, that this work could be done through internal funds or was sufficiently economical that the institution could pay for it themselves. I remember one of the people on my committee at Cornell, when he applied for and got a grant from the National Science Foundation to do his work on bees, the dean called him in and sat him down and kind of say, "What's wrong with you? Why did you do that? Don't you think we can support you?" Well, the world has changed entirely now, where everybody who's hired, and you're going to see this in the Department of Entomology as they are going to be interviewing for positions very soon. One of the prerequisites, one of the expectations is that the person will come in and get an extramurally funded program of research. That is, they're going out and getting grants from the USDA, from the NSF, from NIH, from private foundations, whatever. So the world has changed in that. But the amount of money hasn't grown so fast, and the emphasis on alpha taxonomy has dramatically decreased. You can still get funds to do that, but not to do only that. That is an impossibility these days. And then finally, well, the list probably goes on, but the final thing I want to mention here is the increased emphasis on impact factors in journals. The journals that publish the results of taxonomic research, if they have an impact factor of 1, that is very high. In order to get to the impact factors that some people need for their jobs, especially in countries like China. I have a former graduate student who, to get a job in China, he's got to have X number of papers in journals that have impact factors exceeding, I forget what it was, 4 or something like that. That's not where basic taxonomic research is published. And so even if you are successful in getting a job, the fact that you're not publishing in those journals means that perhaps you won't be kept on. And also in many countries, the grant applications are judged on the basis of what the impact factors were for the work that you've previously done. So all of this this is kind of feeding upon one in a positive or negative feedback loop. I think it's a positive feedback loop, but it's going in a negative direction. It means that there are significant gaps in our ability to identify things. And of course, identification is the key to so much of the other things that we want to accomplish. So how do we address this? Well, here's a couple of papers, a couple of slides showing papers with a proposed solution to that. This one was published a few years ago. I'm looking for the year. I can't remember it off the top of my head. I think it was only like 2 years ago. By colleagues, I know most of these people. A turbo taxonomic study proposing ways to get past the taxonomic impediment. And the method they propose to do it is CO1 barcoded specimens with rapid descriptions of all the species. This was an initial study. They described 179 new species in one paper. Look at the abstract there. You can see all those new species there. Well, the lead intellect here, I think I'm going to put the blame on Mike Sharkey down here. He's just this year come out with yet another paper with the same idea. And instead of 179 species, this one has 403 new species and 11 subfamilies with what they refer to as minimalist revision and description. What that means, let me show you one of these descriptions. Bingo, that's the whole thing right there. A species description that, remember that one of the requirements of the code is that the author provide characteristics by which the new species can be distinguished from other species. And Mike's assertion is that this species, represented by this single picture, you could blow it up, but it's not a great picture to begin with, can be identified by this barcode. Okay, so that gets us into the field of DNA barcode. So let's look a little bit at that. So the idea, you read some of the early seminal papers on DNA barcoding, the justification for it, the idea behind it. The idea of a barcode being kind of like the barcode you have when you go shopping in the grocery store and you run the what do they call them, UPC codes past the scanner in order to get the metadata about the specific metadata, meaning the data about the data. And so the idea was, wouldn't it be nice if there was something like that Mr. Spock could take his tricorder and aim it at a specimen, and the tricorder would be able to tell us what that species that specimen belonged to. So the idea was that the ideal would be to find a stretch of DNA in the genome of the organisms, not too short, because you need some variation in order to if this is going to be a universal identifier barcode, and we have to deal with 1, 2, 10, 80 million species, then we need enough DNA to have enough variation to distinguish everybody there, but not too long, because we don't want to be we want to be efficient. We don't want to be sequencing stuff just for the sake of sequencing it. So we want this kind of sweet spot, Goldilocks length of DNA, and it's got to be DNA. Again, if this is going to be a universal identification mechanism, it's going to have to be DNA that all organisms ideally have, for which we could design some universal primers that you could just go to your favorite primer shop online and say, well, I want the universal primers for the barcode region, and these have been designed, and they are widely applicable. I will give them that. Universal, we're not there. That's for sure. At least for most efficient DNA amplification. As I said, sufficient variation. to be able to distinguish species, but not so much variation within a species that we can't tell if it's one species, whether the variation we're seeing is intraspecific or interspecific. So these criteria have been claimed to have been met by a region called the Fulmer region of mitochondrial DNA, and specifically about 600 base pair stretch of the cytochrome oxidase 1 subunit found in the mitochondrial DNA. And that has been identified as the sweet spot, the sequence to use for DNA barcoding. And this idea has been, I'm trying to find kind words, strongly promoted, flogged by a professor at the University of Guelph in Ontario named Paul Hébert, who has pushed this idea, pushed it very strongly, pushed it internationally, has been talking all over the world about this, founded an institute in Ontario for the storing the barcodes. They have mobile barcoding units, vans specially designed. You go out and collect material on a survey, bring specimens in. You can be extracting the DNA right there in the van. There's been a huge amount of publicity around this effort. I will give Paul Hébert ample kudos for awesome salesmanship of this idea of DNA barcoding gets into one of the papers you read, some of the criticisms, at least for the process of identification of organisms. And one of the positive aspects is that the skills needed to extract DNA to interpret the results that come back from a DNA sequencing center are transferable across all organisms. I could give you a trace file or a sequence file from one of my little wasps, and you would interpret it the same way if you were an expert on plants as I would with the expertise on the group. It's fully transferable. The little bits about nationalism and hubris, the nationalism comes in because the Canadian government has invested heavily in this idea of DNA barcoding, and now it's an international sort of phenomenon. The hubris comes in the idea that that there is a magic bullet, a single answer to this taxonomic impediment that we confront. So you read the main things I really want you to take about DNA barcoding, but let's just hit a couple of highlights here. So here's basically their pipeline. You go out and you collect some specimens. You record the data about the specimens, where it was collected, who was collected by whom, when, where it was collected, who collected it, when, and what you think it is. Then let's take a picture of the specimen to document it, and hopefully, then the specimen will also go into a collection so that you can go and look at it later on if you want. DNA extraction, and usually, at least for insects, what they claim is that all they need is one leg. The insect's got 6 legs, 2 on each side. Sacrifice one, that's gone, but you've got the other one to compare it with always. And there's enough DNA in there to extract this. To extract in order to be able to sequence in this next step the Fulmer region. We're helped by the fact that we're using mitochondrial DNA, so each cell will have multiple, maybe hundreds of mitochondria, so there's a lot more of it than nuclear DNA there. So I'm sorry, we didn't get to sequencing yet. That's the PCR to amplify the DNA. Now we're down to sequencing here. and then we upload it. Make sure that what we have is insect DNA and not bacterial DNA or human DNA or anything along there, by comparison with known sequences to make sure we get close, and then it's databased and available. So that's the basic protocol to get all this stuff identified. And this database will have sequences from other specimens that have been identified, hopefully authoritatively identified, such that if your sequence from here matches what's in the database, then you have an identification there. So that's the protocol. One of the early examples that purported to show the value of DNA barcoding, and an example that has received, well, you read about criticism in the, let me see, who were the authors? Rubinoff, Cameron, and Will. But here, one of the early examples was a paper you read on the genus Astraptes and the skipper butterflies in Costa Rica. That paper, the real motive force behind it, probably both of these people, but I've only put Dan's name up there. This is Dan Jansen, who is an ecologist. And I believe I talked about him before, that he works half the year at the Área de Conservación Guanacaste in a deciduous forest, tropical deciduous forest in Costa Rica. Dan came up with the idea at one point of an all taxon biodiversity. He wanted to focus on Guanacaste and say, he's concerned with conservation there, Área de Conservación. How can we conserve? Why should we conserve something that we're totally ignorant of there? And he really pushed and got a lot of funding for an effort to conduct what he called an all taxa biodiversity inventory, or ATBI, of Guanacaste. He then exported that idea around the world. There was a big effort to do this in the Smoky Mountains at one point. I think it's still nominally going on, and it's been done in other areas. Basically, though, I have to tell you that an ATBI has never actually been done anywhere on any scale. Another thing that Dan was very prominent in promoting was the idea of parataxonomists. If we don't have enough taxonomists around to do all of this work, let's take advantage of local people who, just as a paralegal or a paramedic has some skills in the field but aren't full-blown lawyers or doctors. We can take advantage, we can leverage the labor there, compensated of course, to further the process. Things like doing collecting, doing initial sorting, which means identification, maybe not to the level of species, but pull out the skipper butterflies, pull out the geometrid moths and things like that. And so that's been a big effort there. Also, Dan is known, and you read about this in a couple of the papers, the so-called Caterpillar Project, where he's trying to survey not only what are all the Lepidoptera that occur in Guanacaste, but what do they feed on? Document their immature stages. Document all their parasites and predators. I don't know that he's gotten into viruses and bacteria. Would not surprise me. But he's definitely gotten into the hymenopteran and dipteran parasitoids that are associated with all these butterflies, to build up a comprehensive picture of what these LEPs are doing in that environment. So that's Dan. This is Dan here. This is his wife, Winnie Hallwachs, who is also Dan's the big spokesman. He's the charismatic half of the marriage, but I cannot think of a single paper that has ever been published with Dan's name on it that Winnie was not right there with him, at least during the times that they've been married. The Astraptes paper is commonly put forth as an example of how DNA barcoding can reveal to us that what we thought on the basis of external morphology was a single species, and in the case of these butterflies, single species whose larvae fed on a wide variety of different plants, that they were generalists. When you look at their CO1 DNA sequences, it's revealed that no, we don't have a single generalist species. We have several specialist species. So in fact, the ecology combined with the CO1 barcoding is telling us that there's more than one species there. And in this case, from 1 to 14, there are other even more dramatic cases out there about, especially the example of the tachinid flies and Braconnid wasps. That second paper I showed you of Sharkey, where they described over 400 species. That was all of those specimens came from Dan and Winnie, them rearing them out of their various caterpillars over the past probably half a century that they've been working in Guanacaste and recording all of this information. So there's Dan in the background. I'm not sure who that is there. I don't recognize her. But, that's a pretty extravagant, that's not one of the Astraptes. These are the Astraptes caterpillars. That there's a lot of diversity in the color patterns and the colors of the larval stages. The adults pretty much look all alike. But a lot of that diversity was thought to be just represent that we have a species that there's a lot of different colors in the larvae. But when you combine what they will feed upon and the DNA sequences, you see that we have many more species there than we had any idea or any hope of recognizing before. So this is played up as an example of DNA barcoding can not only help us to identify known species, but it can help us to identify undiscovered species. And the criticism of that is what is in the paper by Rubinoff et al. One of the things inherent in the use of DNA barcoding to distinguish species is this idea that if we took a random, let's say we took a random sample of specimens, let's say 100 specimens, and we sequenced the CO1, the Fulmer region of them all. And then just as you did in lab, did a distance matrix of this, first of all, you align the sequences, and that's a critical point that many people gloss over. And then you calculated the distance between every pair. You did a pairwise distance matrix, just as you did. So every pairwise combination of your specimens, you calculated the distance and then graphed that. So this is the distance represented here as percentage. So the percentage difference between the sequences you're looking at. And this is the frequency of specimens that had those differences. So obviously this is more than 100. I should have counted that up. But the idea is that if you're comparing different species, then you're going to find relatively big differences in their CO1 sequence. And if you're looking at two specimens that are the same species, their CO1 is going to be much more similar, very close to one another, maybe only a few nucleotide differences. And if you graph this out on a frequency as a frequency distribution, we would see a bunch of specimens that are very similar, and then a bunch of specimens that are, or specimen combinations, I should say, that are much more different. And that there is a gap between these two, the so-called barcode gap, that differentiates for you intraspecific variation from interspecific variation here. In this case, this barcode gap looks to be about from 2.5 to 4.5%. So you would use this data if you empirically determined it for the group of organisms you're looking at. You would use this data to say, if I compare two specimens and they are more than, let's say, 5% different, then these two specimens belong to different species. If I compare these two specimens and the difference in their sequences is less than 2%, that's the same species. This number empirically, though, seems to vary a bit. And it's commonly cited that a difference of 2.5 to 3% difference is a good threshold for distinguishing between species. In the Sharkey et al. Paper, the 400 new species from Guanacaste, from Costa Rica, that I flashed by you there, they use a threshold value of 2%. They did not empirically determine that 2% was appropriate for these taxa. And remember, they were also looking across, 11 different subfamilies. So I would have expected some variation in that number across those different groups. But they use that as their threshold. And any specimen that they got that was more than 2% different from other things, any other things, they called a different species. And for that one species whose picture I showed you, that was a 594 nucleotide barcode. A 2% difference would be about 12 nucleotide difference. As a systematist, I would have to ask myself, first of all, how much variation is there within a species? Is it limited to the 2%? Or perhaps is it more? Secondly, I have to add in variation in the sequencing technology. Perhaps if you're using, as that initial workflow showed, individual PCRs and Sanger sequencing, the error rate that you would expect from that is going to be low enough that it's not going to substantially impact numbers at this scale. But you get higher error rates in the newer techniques that people are using, next-gen sequencing, Illumina sequencing, to do this en masse. And that's got a higher error rate, and that may impact this. So in these 12 nucleotides, how many are different just because of differences amongst individuals, different haplotypes, if you will, existing within the population? How much to technological error rate? And at least in the Guanacaste example, they were looking at one place. The Rubinoff et al. Paper pointed out that when you look at things with wide geographic distributions, and you only look at the 2 points farthest apart in that geographic distribution, they are likely to exceed this 2.5 to 3% threshold. So it's not a simple, straightforward, can I say brain dead, mindless? These are not good adjectives to be using here. There are issues that are involved in this, using these thresholds. The first question to ask, of course, and a question that is frankly rarely asked, is does a DNA barcoding gap exist? Or does the amount of variation within species and between species intergrade such that you're not getting a clear signal of the difference in this single sequence? There we go. So I guess I like all the papers in the sense that people are putting forth their position in the strongest manner they can, and hopefully letting the inherent value of their arguments play out. I've talked to you about, Jansen and Hebert have been very, very, very vocal proponents of DNA barcoding for identification, for discovery of new species. You had one critical paper there, Rubinoff, Cameron, and Will, who, by the way, Dan Rubinoff works on Lepidoptera. He's now in Hawaii. I guess he was on that paper too. Steve Cameron is an Australian who is now the chairman of the Department of Entomology at Purdue. And Kip Will is an Ohio State entomology alumnus. He got his bachelor's degree here at Ohio State, and he's now a professor at Berkeley. So they voice a number of criticisms, some of which I've mentioned. The others I'll leave it up there, up for you to read about in their paper. But there are other issues too that they only that perhaps they didn't talk about. What if, for example, the formal reason doesn't work? There are in fact some groups, I believe Nidaria are one of them, where the Fulma region, they have mitochondrial CO1, but the Fulma region basically shows zero variation. And so that's not going to be useful as a DNA barcode for them. So they have, the people who think this is nevertheless a good idea, have searched for and proposed alternative candidates. So we're getting away from a universal identifier region for animals. The plant people, CO1 doesn't work. They use a different section as well. So again, well, hopefully most of us can distinguish insects from plants, but you're going to have to tailor which DNA you barcode if you subscribe to the value of DNA barcodes for identifying new species. You're going to have to tailor what you're looking at. For my 2 cents, come down strongly on the side of what Rubinoff et al. Are proposing, is that the DNA sequence in the Fulma region, the barcode region, is valuable data. It's data that's useful. It's data that should not be eschewed, should not be tossed out, but it's data that should be used in combination with other sources of data. With, for example, nuclear DNA sequences. What we've used is to combine CO1 data with ITS data, so excuse me. It's a sequence that occurs between RNA copies. I can't remember what the ITS stands for off the top of my head. Combine that with morphology, combine that with behavior, combine that with ecology, combine that with distribution. All of these things need to come together to be integrated so that we see how all of these things play together in order to tell us what species are in some of these groups that are very difficult to identify, to distinguish. I don't think that the example papers I had up there from Rakanitz, that the complete reliance on CO1 to come up with your species hypotheses is an appropriate way to do this. I don't think that coming up with quick but flawed answers to how many species there are and what names we put on them is an effective way to address the need, and this is the need that Jansen speaks of with great emotion and urgency, the need to inform the general public of what's out there in the world so that we can preserve it. Personally, I don't think that coming up with a bad answer is the way to convince people that they need to invest in these types of studies. And ultimately, I think it also leads to a reprise of the whole taxonomic impediment problem. In order to identify them, you need authoritatively identified specimens that are sequenced. Who does that? The taxonomist does it. If there aren't taxonomists, who does that? And further, if it takes no particular taxonomic or expertise in a taxon, let's put it that way, to get a DNA barcode sequence, and frankly it doesn't, then why do we need taxonomists? Let's get rid of the taxonomist. So instead of alleviating the taxonomic impediment, it leads to a further decline in the job opportunities and funding for taxonomists going forward. So I fear that it could, in the long run, be bad for taxonomy in systematics. If I can find my way to there we go. So, in summary, dichotomous keys are a standard tool for identification, and identification is what you really want in the end, and everything else, even my pedagogical needs, should be considered to be secondary. That's not to say that things like multi-entry keys don't have a place, don't have value. They can, and the two worlds can be combined. You can create from the same data set, create both a dichotomous key and a multi-entry key. It doesn't have to be one or the other. It can be both. The taxonomic impediment is a real thing. Expertise in taxonomy is declining. The number of students being trained is declining, and there are still vast numbers of species about which we know absolutely nothing. And one thing Jansen's right about is that if the public is going to invest in conservation and preservation, then they need to be told what the value is that they're going to get out of that, other than just some aesthetic value. I think DNA barcoding has definite use in the context of taking an unknown specimen and matching it to a known specimen by using this sequence. Whether it has value in identifying new species is something I'm less sanguine about, although I do think that it has a place in a program of integrative taxonomy to be bringing all information from all possible sources together in order to come to a scientifically robust hypothesis about what the level of biodiversity is in the world. So I'm going to stop now for this, and we'll catch you next time on your same bat time, same bat channel. Take care, stay healthy.